All right, in this video, I'm going to show you exactly what it is I want you to be able to do and what I want you to know regarding the four complexes of the electron transport chain. And so I'm just going to go draw them out as I would want you to be able to draw them out. And we're going to start with complex 1. So I'll write that up here. Complex 1. And of course, complex 1 is a transmembrane protein, and it spans the inner membrane of the mitochondria. So let's go ahead and do that. Let's start up here. So there. And then we have, of course, there's our complex 1. And then let's continue like that. So that what we have here is this is going to be the intermembrane space. Membrane space, and this is going to be the matrix. So make sure that you always know your compartments when talking about this stuff. And then this is the inner membrane of the mitochondria. Inner membrane. So in complex 1, what we have is the oxidation of NADH. So NADH becomes NAD plus a hydrogen that ends up in the matrix, a proton that ends up in the matrix. Now that NADH, right, is going to reduce Q, quinone, ubiquinone, excuse me. And so ubiquinone becomes completely reduced. Now, the source of those two hydrogens come from the matrix. So let's go ahead and draw that like this. So 2H plus from the matrix. Okay, so notice we're only getting the H pluses from the matrix. The electrons are coming from our NADH. And so we'll write that right like that. Okay, so we have an exergonic redox reaction in which NADH reduced Q to become QH2. This is an exergonic reaction, which means it released energy, and that energy can used to do work. All right, what work was done? Well, we took 4 protons from the matrix and pumped them into the intermembrane space. So basically, and this is, right, we're pumping protons against their concentration gradient. We've got a higher concentration here in the intermembrane space and a lower concentration here within the matrix. So that's an endergonic reaction. Pumping protons against their concentration gradient is an endergonic reaction. So essentially what we've done with complex 1 here is we've coupled 2 reactions. We've coupled the exergonic reaction of this redox reaction of NADH reducing Q to QH2 to the endergonic reaction of pumping these 4 protons from the from the matrix to the intermembrane space. But now let us write out a balanced equation for this reaction where we started with 6 protons from the matrix, and we have ended making a proton in the matrix. So that is a net of 5 protons from the matrix. So what we start with then is NADH plus 5 protons from the matrix, and that's going to yield NAD plus, oops, we almost forgot, plus Q. We need our oxidized Q. So I'm sorry, but it's going to look a little sloppy, but we'll write that There. So we have our oxidized NAD plus our fully reduced Q, QH2, plus our protons out here, 4H plus in the intermembrane space. All right, so let's count up our hydrogens and make sure everything fits. Well, that's 4, 5, 6. Okay, good. So 6. And that's 1 plus 5 is 6. Fantastic. We've balanced everything well. NADH has become oxidized and Q has become reduced. So now the next step is to figure out eventually what happens to the electrons being carried by Q. But before we do that, we have to look at complex 2, because in complex 2, it's another way of doing this. Complex 2 represents another way of reducing our Q to QH2. So we'll look at that next. Okay, so the next enzyme within the electron transport chain is complex 2. Now, complex 2 is better known as succinate dehydrogenase. That's the same succinate dehydrogenase that exists within the citric acid cycle. So we have an overlap between these two mechanisms. This overlap is succinate dehydrogenase. It's complex 2 within the electron transport chain, and it is one of the reactions of the citric acid cycle. So like any complex within the electron transport chain, it is embedded in the inner membrane of the mitochondria. So here's our inner membrane, and let's make it extra big this time because there's a lot we have to account for. We'll make this a little bit wider. And we'll continue on here. So to get our bearings, this is the intermembrane space. This is the inner membrane. And this is the matrix of the mitochondria. And this is complex 2, or succinate dehydrogenase. And what we have here is we have succinate is our source of electrons and hydrogens. And that is going to initially, its electrons are going to be taken taken by FAD. There's FAD. Remember, FAD, unlike NAD+, is a catalytic cofactor, meaning that FAD is covalently linked to succinate dehydrogenase. It can't go and come as it pleases like stoichiometric cofactors such as NAD+. It's stuck there. That's why this succinate dehydrogenase has to be an integral part of the electron transport chain. So when succinate donates its electrons and hydrogens to FAD, FAD, of course, becomes its reduced state here, FADH2, and succinate, when oxidized, becomes fumarate. And now remember also that the reason we're using FAD here, not NAD+, is because what we're doing here to oxidize succinate is we're not converting a hydroxyl into a carbonyl, we are converting an alkane into an alkene, a carbon-carbon single bond to a carbon-carbon double bond, and that demands a higher, something with a higher oxidation strength, like FAD. Now, of course, right, we're not done here, because the important part is to transfer those electrons here off to something that can move to the next complex, to complex 3. And of course, FAD can't do that because it is covalently bound to the enzyme here. So what we have, of course, is our Q. Q comes in. and becomes QH2. Where does it get those electrons? Where does it get those hydrogens? It gets them from the FADH2 that stole those 2 hydrogens from succinate. So our balanced equation here, oh, by the way, even though this is an exergonic reaction, it's not being coupled to the pumping of protons here. We're not moving any protons from the matrix out into the intermembrane space. So our balanced reaction is going to look like this. Succinate plus Q gives us fumarate plus QH2. Where do these 2 hydrogens come from? They come from succinate. Okay. And now we have this accumulating pool of reduced QH2. We've got some from complex 1 coming from NADH, and now we have some from complex 2 coming from succinate. And now we need to do is look at where does this QH2 take its electrons? What is the next redox reaction that is going to occur? And that'll occur in complex 3, and we'll look at that in just a moment. Complex 3 is one of the trickier ones. It is the one in which that QH2 is going to be oxidized and cytochrome C is going to be reduced. So again, like all of the other ones, this is just a redox reaction that is exergonic, and it is going to be coupled with that endergonic reaction of moving protons against their concentration gradient. And this one, the reason it's tricky is because we have to take use of something called a Q cycle. And the ultimate problem here is that Q holds on to 2 electrons and cytochrome C only holds on to 1 electron. And so there's a very specific way that this has to occur in order for us to transfer these electrons from Q to cytochrome C. So let's take a look. What we're going to do is we're going to draw 2 snapshots of complex 3. So here's snapshot 1. All right, and let's go a little bit further, a little bit further, a little bit further, and then here is snapshot 2. So it's the exact same enzyme, it's just 2 points in time here. Of course, we have our intermembrane space here. This is the matrix, and this is the intermembrane. Be careful, this is the inner membrane with 2 ends. This is the intermembrane space, not inner membrane space. And we are taking and oxidizing QH2, and the oxidizing agent, the thing that's going to be reduced, is cytochrome C. Okay, so this is the way it works. 1 full QH2 becomes oxidized. One of its 2 electrons goes to cytochrome C. Another one of its electrons, remember it has 2, can't go to cytochrome C because it can only hold 1, so the other one goes to a fully oxidized Q that becomes partially reduced. Okay, and then the two protons here go into the intermembrane space. H plus intermembrane space, and this is 2 of them. Now we're in a second snapshot where our number 2, QH2 number 2, comes in, goes through, and it becomes fully oxidized to Q. Same thing. We have now. This is number 1, cytochrome C number 1. Here's cytochrome C number 2. Number 2 site. Oh, let's erase that there. Number 2 site C. So our 1 electron goes over there, And one electron, then our two hydrogens are pumped out into the intermembrane space. And the second electron goes to fully reducing that Q to QH2. Of course, we need a source of the two protons. So we have one electron here, we have another electron here. We need the two protons to balance that out. Those two protons come from the matrix. All right. Okay, let's balance this out. We put in 2 QH2s, but 1 came out. So the net effect is we only put 1 in. So let's start way over here. So that's QH2. We put in 2 oxidized cytochrome Cs. So plus 2 cytochrome C oxidized. Okay. And then we also put in 2 H pluses from the matrix. Plus 2 H plus from the matrix. Okay. Now, what did we get out of it? Well, let's take a look. We got out of it our oxidized Q. I'm going to erase this here because you know what it is already. So our oxidized Q, our 2 cytochrome C reduced, and plus 1, 2, 3, 4, 4 H plus intermembrane space. Let's make sure that that's all balanced. 4H plus, 1, 2, 3, 4. We're good to go. So this is complex 3, the Q cycle. Take a look in your book at the figure that's happening here. Take a look at the video that I have posted regarding this. It is a little bit tricky to understand. I want you to be able to do what I've just done here. The last complex, the last place that these electrons that have started with our reduced fuel source, like glucose or a fatty acid, the electrons that are traveling here in the 2 cytochrome Cs, where are they eventually going to go? Well, that last acceptor, electron acceptor, is oxygen. And the enzyme that catalyzes the transfer of electrons from cytochrome C to oxygen is complex 4, and we'll look at that next. All right, complex 4, the final one. Complex 4, like all the other complexes, is a transmembrane protein that exists in the inner membrane of the mitochondria. So let's make it nice and big here. All right. And what it does is it takes the electrons from 2 cytochrome Cs, 2 Cyt C that have been reduced, and it oxidizes them. So 2 Cyt C oxidized. And the oxidizing agent is the absolute epitome of oxidizing agents in that it is oxygen. So in this case, oxygen, because it oxidizes cytochrome C, oxygen becomes reduced. What does oxygen become when it becomes reduced? It becomes water. So we have 1/2 molecule of oxygen. All right, grabs those electrons. And we need the hydrogens, right, to balance out those electrons there. So we have 2 protons from the matrix that go into this. And then From that, we have our reduced oxygen, which is water. This is an exergonic reaction like all of the other reactions of the electron transport chain, and this one, like reactions number 1, like reactions number 3, this one is coupled to the pumping of 2 protons from the matrix to the intermembrane space. Okay? Nice and easy, right? This is the reason we breathe, right here, folks. Oxygen has the highest reduction potential. It is the best oxidizing agent. It is the reason we breathe. We breathe in so that it can pull all of those electrons ultimately from our reduced fuel source through the electron transport chain to oxygen to become water. We excrete the water and we breathe in more oxygen. Now, let's balance out this reaction though. So we start with 2 cytochrome Cs that are reduced. We add in half an oxygen molecule. And we add in 4 H plus from the matrix. And what do we get out of it? We get out of it our 2 site C that are oxidized, plus 1 water, plus, whoops, that's 1 water, plus our 2 H pluses that we've pumped into the intermembrane space. 2 H plus intermembrane space. Let's balance those. There's 2 hydrogens in the water. There's 2 hydrogens there, the 2 protons. 2 plus 2 is 4, and that is 4 right there. All right. So at this point, the traveling of our electrons are done. They started in a very reduced fuel source, such as glucose or fatty acid. They have traveled along with NADHs, or perhaps they were taken straight to the electron transport chain via succinate dehydrogenase. They were passed along, involved in several redox reactions until the final one here where they end up with a molecule of water. And in so doing, where's that energy now, right? Where is the energy of those electrons that we got from our reduced fuel source? They are in the proton concentration gradient that we have made. And the name of that energy, the name that we give the energy inherent in this proton concentration gradient that exists in the intermembrane space, we call it the proton motive force. And the question is, what does the cell use this proton motive force to do? Well, of course, you know the answer to that. It uses the energy inherent in the proton motive force to make ATP. And we'll look at that in another lecture.